Hello and welcome everyone. Welcome today to our webinar, part of the Innovation at Bachem series, where today we'll be looking at CEPS for protein synthesis, with some selected examples of complex proteins. So a little bit about Bachem before we start. We at Bachem are one of the world's leading peptide manufacturers. We are a contract and development and manufacturing organization. We specialize both in peptides, but also more recently in oligonucleotides as well. We have long-term partnerships within the pharmaceutical industry and with small and medium-sized biotech companies. And a little bit about who we are. So I'm Seamus White. I'm a business development manager, and I work for Bachem Bubendorf in Switzerland. And I'm very pleased to introduce Leendert from the CEO and co-founder of EnzyTag, who will be giving today's webinar. A little bit about the housekeeping before we get into the webinar itself. At any point in time, you can ask questions via the Q&A section at the bottom of your screen. There won't be any, you will be muted throughout the webinar, but please feel free to ask questions at any point. We will get back to these questions at the end of the webinar, and questions are anonymous. So a little bit of an introduction about Bachem. As you can see, we're split into 3 different segments. So we cover the full spectrum from discovery and research and custom peptides at the early stage, non-GMP, through CMC development. And then later we move into commercial, so generics and NCEs. And we also have our own portfolio of generics peptides as well, and also oligos. And where are we situated? Bachem, it's headquartered in Bubendorf in Switzerland. It has here on the world map, you can see in orange are the GMP sites. So we have a presence on the west coast of the US at Torrance and Vista. And also we have a custom synthesis group in St. Helens in the UK, 2 sites in Switzerland, and a sales office in Tokyo. So we are specialists in large-scale API manufacturing. We have the full range of solid phase peptide synthesis capabilities. We have closed and contained peptide isolation facilities, wide variety and a vast array of chromatographic techniques, and also large-scale prep HPLC capabilities. So this webinar is actually a continuation of our earlier webinars on CEPS, so Chemo-Enzymatic Peptide Synthesis Technologies. We had one back in March where we had the introduction, and then in May we had the Peptiligase Selection and Engineering webinar, and all of those are available on our social media channels. And today I'm very proud to host EnzyTag for this final webinar in the series. So over to the main person today, Leendert, would you like to take control?
Thanks a lot, Seamus. It's much appreciated. And I hope the sound is fine so that everybody can hear me.
Yes.
Yes. Okay, thanks a lot. So it's a great pleasure again to participate in the webinar series and discuss more on CEPS for protein synthesis this time. So indeed, the 3rd in a row this year, we've collected a couple of examples on protein synthesis that I would like to talk you through, because it's CEPS for protein synthesis. And I think this is important to start with. There's an overview of the peptides and proteins that are currently in scope of pharma companies. So to start at the far left, which are the short peptides, and that are of course very well known to the Bachem community, because these are produced a lot by Bachem. And then you have the GLP and the GIPs, which are already longer. So they are in the range of 40 amino acids, sometimes a bit larger, sometimes a bit shorter. Then we have insulin, which is a special case because that is mostly produced recombinantly. Then we have various others, which are relatively large, so starting at 50 and ending in the hundreds. And then we have a couple of special classes like chemokines, non-antibody small protein scaffolds, cytokines, and immunoglobulins. So the pharma industry is focusing a lot on those substrates. Most of them are engineered, modified, or conjugated to extend the half-life, or for other means, they are modified for other means. And on the next slide, we have an overview of what is produced by what means. And to again start at the far left, which is the short peptides, the GLP and the GIPs, those are readily produced by solid phase peptide synthesis, and everything more or less until 60 is produced without any problem. Of course, it is sequence dependent and case by case could vary. Then we have at the far right the recombinant ones, where you have the real large proteins that are easily produced recombinantly and could easily be scaled up. And then in the middle, there is a class of peptides, or mini proteins, as you would call them, that are quite difficult to get to by solid phase peptide synthesis and are sometimes too small to produce recombinantly, and causes all sorts of problems. Of course, if you'd like to modify them by glycosidation or pegylation or lipidation, that is of course not possible using recombinant, and that is where we see fit for our chemo-enzymatic peptide synthesis technology, or chemo-enzymatic protein synthesis technology. And to just a little bit on the technology itself. So here it is, it's enzymatic. So peptiligase is at the heart, and the peptiligase is a patented technology where a couple of, so it starts, the basis is a subtilisin enzyme, and a couple of modifications have been applied to that subtilisin enzyme. Subtilisin itself is a protease, and it's a broad spectrum protease, so it does not require a unique sequence of amino acids to clip, to hydrolyze. And what has been done is that the serine in the active site that is causing the hydrolytic properties has been replaced by a cysteine. And in that way, the protease was converted into a ligase. So instead of cleaving peptide bonds, it ligates peptide bonds. And in addition, because by that change to cysteine, the enzyme loses a bit of stability. So what has been done in addition is a couple of stabilizing mutations in the backbone of the enzyme that improved the activity of the enzyme and also made it a bit more stable to, for example, some percent of organic solvents, and also some chaotropic agents are tolerated. So here is then how it exactly works in the enzyme. Sorry, I'm just working myself a bit through the technology here. So there is the enzyme, so at the heart, and then you have the cysteine sticking out of the active site. So to that enzyme, we feed an ester fragment, which is obtained by solid phase peptide synthesis. The ester fragment, the orange colored fragment, has at its C-terminal an oxoester. And what happens is that the cysteine of this, the thiol of the cysteine attacks the carbonyl over here, forming a thioacyl complex, which means that the ester fragment, the orange fragment, is covalently attached to the enzyme. And the alcohol leaves, so that is the blue block over here. Then the amine fragment comes in and replaces the thioacyl complex, forming the native amide bond. So, and the product itself has just an amide bond, like all the other amide bonds that are produced by solid phase peptide synthesis. So in that way, it is without any scar, so without any traces left of the enzymatic ligation. And here is an enlargement of the active site. So the enzyme recognizes 6 amino acids positions, 4 in the ester fragment, which are colored orange, and 2 in the amine fragment. And the specificity of the pockets is also depicted over here, with S4 being the most specific in that it prefers hydrophobic amino acids, but not just one like phenylalanine, but the whole range of hydrophobic amino acids is accepted in that pocket, and sometimes even other amino acids like tyrosine. Then S3 is a very shallow pocket, so it essentially only recognizes the amide backbone because the side chain is solvent exposed. S2, S1, S1 prime, and S2 prime have medium specificity, but we know what amino acids make up that pocket, and we could engineer certain variants that do prefer certain amino acids in that pocket. So either bulky amino acids or charged amino acids or small amino acids. And this has been done already. And we have these enzymes available off the shelf in EnzyTag. Even non-natural amino acids are tolerated. So it's not necessary to have completely canonical amino acids between or in the S4 till S2 prime. So you can also play around with D amino acids in certain positions. Modified lysines are tolerated, and sometimes even alpha methylated amino acids are tolerated. Everything outside of those 6 amino acids is not so much of interest to the enzyme, which means that you could have a peptide, but you could also have a protein, but you could also have another non-natural construct like a nucleotide or a glycoside or a steroid or whatever is of interest you want to ligate. So then back to the to the slide. So here is the enzyme. What has been proven is that we typically do these reactions at a low micromole scale in the lab, but it has also been proven already at kilomole scale in GMP facilities. So there, the Peptiligase, the technology itself is intrinsically scalable, and because it's an enzyme and it runs in aqueous environment, it is environmentally friendly. So the compound, the peptide you obtain as the product has a higher purity, and in that way it also requires less purification afterwards, and so you also reduce your acetonitrile consumption. Then on the next slide, this is essentially the summary of what I would like to present to you in the remainder of this webinar. To just walk you through a couple of examples where we apply chemo-enzymatic peptide synthesis for the synthesis of proteins. And I do that using a couple of examples of very long peptides or small proteins. I will use these interchangeably during the webinar. And so we typically start when we get the sequence with identification of the ligation point. We have several means for doing so, and I will tell you what means. Then the second step is that we obtain the fragments via solid phase peptide synthesis. The third one is that we do the ligation development. And the fourth one is that we do a cost-efficient upscale under GMP at Bachem. So we are a CRO. We have lab facilities in the Netherlands, but we have no GMP facilities. For that, we partner with Bachem. So here is the first example, which is glycine-rich protein, GRP. That was a request we got in from an academia in the Netherlands that wanted to have this protein for screening purposes. They failed in making this by solid phase peptide synthesis, and also native chemical ligation wasn't an option because there was no cysteine present in the sequence. And what we did is we got the sequence, and we have an algorithm running in the lab that quickly identifies ligation points, and that's what you see here. So we feed the sequence to the algorithm, and we get a list of possible ligation points out of it. And here you also see the scope of what the enzyme can do, because you see many ligation sites in just this sequence. It's not a random sequence, of course, but there are many sequences like this, of course, in the databases. And so the first three are denoted, and they have a box around them. So in all cases, it's 6 amino acids. So this is number 1, this is number 2, and this is number 3. And the score was reasonable to good. So I realized the score don't say you a lot, but we know how to interpret these numbers. And of course, in order to benefit the most of solid phase peptide synthesis and then ligating 2 relatively long fragments, we decided to go for the ligation site, which was more or less in the middle. And here you see how then the fragments look like, with again the ester piece in orange and the amine part in green. What we then do is we synthesize the fragments using solid phase peptide synthesis, and then start testing the ligation. As an interim step, we sometimes test the ligation site for model fragments. So then we don't take the full-length fragments as depicted on this slide here at the bottom, but we synthesize fibers, so model peptides with the correct sequence for the ester and the amine, and just see how fast the conversion is. And I included this slide to give you an idea how fast the enzyme is. So we synthesize these fibers, and here you have the ester, and here you have the peak of the amine. The amine is added in a bit of excess, and we added the enzyme together in a buffered solution. And here you see them almost in, well, 60 minutes, but at 30 minutes, there's quite a bit already converted in the product. And yeah, as you see, we let the reaction stand for a little bit longer to just see whether there's any impurities formed or whatever, and that is not the case. You see a bit of hydrolysis, and that is because the ester is base labile. So that's also already present at the start. That is because the pH we do the ligations at is 8.5. So we sometimes see a bit of background hydrolysis or chemical hydrolysis. And the other contributor to hydrolysis is enzymatic hydrolysis. That's quite a bit of water present in the active site of the enzyme. And although the amine, reacting with the amine is the preferred pathway, sometimes it happens that water attacks and gives hydrolysis, leading to the ester starting material. So knowing that we get quite efficient conversion into the product, we took the real fragments, so we synthesized the real fragments and we treated them with enzyme. And in this case, you see here the amine. You see here, so this is already the 120-minute trace. Here is the product coming up. You see a hydrolysis peak and you see an ester peak, and here is a peak of an internal standard, which was not, which is not marked. The amine was added in quite a bit of excess because we had quite a bit of the amine, and we just did one trial because the academic institute was really waiting for the material, and we wanted to just do it correctly in one go. And so after 240 minutes, the conversion went on, and after 360 minutes, it was more or less done, with the product peak being the predominant one. There was still amine present, but that was added in 4 equivalents. So that was no surprise, and we saw a bit of hydrolysis. So then we did a purification, and here you see the product peak, and here is the mass confirmation of the product peak, and also a bit of information on the purity. Here just a bit of numbers. So we synthesized 8 milligram of ester, 14 milligram of amine. We ligated that together and were able to isolate 2.1 milligram of GRP pure and 1 milligram of GRP with a trace of an impurity. And so we were able to do so in 4 weeks' time. So between when we got the sequence from the academic institute all the way to delivering the purified peptide, and so they then could continue with their work on discovering how the mechanism of action was of this protein. So for the GRP, I showed you the way to or identifying the ligation point using the algorithm. Here is a slightly different way of identifying the ligation point, and for that I took EGF. EGF in itself is not a very interesting target because it can readily be synthesized using recombinant expression, but we just took this one because it's cheap, it's complex because it has a couple of disulfide bridges, and we wanted to know whether we could also identify ligation points and then subsequently ligate the fragments together. So for that, what we did is we took the ligase and we installed the serine back in the ligase. So which means that the ligase has all the properties that the peptiligase offers, but then just by simply replacing the cysteine for the serine, it gets hydrolytic properties with all the recognition and docking properties of the corresponding ligase. So what we typically call these is omnilase versus omniligase. So what we did is we took then EGF, we reduced the disulfide bonds. I will just take it from here. We reduced the disulfide bonds first in order to just stretch the complete sequence and then feed that to the hydrolase. And here is what you then see. So you need to be pretty fast because the omnilase, so the protease, is very active. So you need to sample each and every 5 minutes. Here is the unfolded EGF and already at T is 0. So immediately after adding the hydrolase, you see already peaks coming up. And by just carefully reading those out from the MS, we could identify the following fragments in the MS. So at least the complete fragments, because soon, already after 30 minutes, you see that also the fragments start breaking up. And after a couple of hours, you end up with dipeptides or tripeptides as small as that. And we were able to identify a couple of ligation sites, as you can see here. And that is summarized on this slide. So here are the fragments that we could identify. So 3 in total, where we had a fairly good trust in that they are a very good ligation site. And we selected this one, and we subsequently produced these ones by solid phase peptide synthesis. We produced the ester, we produced the amine, and because of the lower solubility of this amine fragment, we installed a ester at the C terminal of the amine fragment as well. So that's then an OH-MBA ester. We know that that ester is less reactive towards the enzyme. And we added a couple of lysines. So we added charge to that fragment in order to solubilize it. And here you see how the ligation went. So at T is 0, I admit that the HPLC is not the nicest, but the ester and the amine are more or less in the injection peak, and then at T is 0 you see already the EGF plus the solubility tag coming up. And here, so you see that grows in about an hour. And then the pH was increased in order to hydrolyze away the OH-MBA ester, and that gives after 60 minutes at pH of 10 to 11, the EGF unfolded. And currently folding studies are going on, but that is all known, and already quite a bit of that is published. So we just follow literature procedures there to get the EGF folded. So we typically recommend these hydrolase studies when the 3D structure, so the secondary structure of the protein is not known. And we essentially then, kind of let the enzyme guide us to the correct ligation site, taking into account secondary structures, compared to our algorithm, which just looks at the peptide or protein sequence and seeks for the hydrophobic amino acids and other characteristics that are well recognized by our enzyme. So I think in that way, the hydrolase study adds an additional level of well, it elucidates an additional level of complexity. And the last example I would like to present to you is one for a customer-sponsored project. I can't disclose the sequence to you, but I can say that it is a very long one. So in this case, 100 plasmer protein where no native chemical ligation site was available, and which had a variable functionalized domain, so the blue part, and a constant domain. And the sponsor was interested in whether we could synthesize various variable domains and then ligate that in a couple of ligations to the constant domain, in that way speeding up their R&D program, their discovery program. So the customer is Affibody. It's a Swedish company that was interested in this and that wanted to test this in vitro and in vivo. So what we did is, because these proteins were not available, we just fed the sequence of interest to the algorithm, and we got various ligation sites out, and we choose one which was located in the constant domain in order to just get the freedom there to ligate it together. We synthesized then the various functionalized domains and of course also the constant domain, and started to develop the ligation site. First small side step again. So because the N-terminus of the variable functionalized domain would have been recognized by the enzyme, and in order to then prevent cyclization or dimerization of just the variable domain, we needed to apply a protective group at the N-terminus. And we have various examples for that. So we have an FMOC or an SMOC, and a bit dependent on the solubility, the overall solubility of the variable domain, we could also just play around with that. In this case, we decided to go for the FMOC, and that went extremely well. We can also go with DDE, which is removed by hydrazine. I know there are also a bit of drawbacks to DDE and hydrazine treatment, especially if you have some glutamic acids or glutamine derivatives present in your sequence. Phenylacetyl protective group could be applied, which can be removed enzymatically. So we've also experiences with that. And we could also, for example, use the Edmond degradation by installing an additional amino acid at the N-terminal, for example, proline, which is not recognized by the enzyme and subsequently removed using the Edmond degradation. So there's not really one best in class N-terminal protective group. So that still requires a bit of thinking and trying before we have the correct protective group. So it also very much depends on the sequence, on the amino acid sequence. So after we've synthesized the fragments, which is not shown, we ligated them together. And here is the example of the crude reaction mixture after 1 hour. Again, the amine was added in excess. Then we have the P of the product, and we have a bit more hydrolysis. And that had to do with I think also because of the length of the fragments, which was fairly large. And of course, it could adopt all sorts of secondary structures in solutions or in our buffered solutions. And so that's why you see more hydrolysis compared to the model fragments and compared to other ligations. And here's after 2 hours, so hydrolysis is a bit lower, product peak is a bit higher, and also the amine is a bit lower. And that's where we stopped the reaction. We didn't see any more conversion, and we then purified the mixture. And that is what is seen over here using a 40 to 50% acetonitrile gradient, and confirmed the identity by MS against a recombinantly produced variant. So this one, the LC I showed you, where everybody was also able to produce it recombinantly in order to just have a benchmark. And with this, the in vitro experiments are completed, and the in vivo experiments are ongoing. So those were the three examples I wanted to show to you about the synthesis of proteins. So where you go well above the 60 amino acids, where yield is then becoming a serious problem. And by ligating longer fragments together, we are now able to go well above 100 for protein synthesis. We can insert non-canonical amino acids and non-peptide motifs at will, because we base ourselves on solid phase peptide synthesis, and that offers that as well. And because we ligate, we use enzymes, we use aqueous solvents. The purity of the products is in general higher to much higher compared to solid phase peptide synthesis. We can also significantly reduce the solvent consumption. So here's again the summary of what we have on offer: the robust technology to manufacture complex peptides and small proteins without non-canonical amino acids and non-peptide motifs, and the amount of solvents is significantly reduced. So thanks a lot, and I think it's back to Seamus now.
So I'm just looking down through the questions. I think some of them are similar in theme. So I'll go with, so how does the cost of a single-use enzyme compare to reusing the enzyme? So immobilization, and that was asked by more than one person.
Yeah. So the cost of the enzyme is not that high. So well, what is that high? Of course, so what the enzyme is produced at the CMO. Titers are high gram per liter, and it can easily be produced in grams, multiple grams. So that is not the problem, which brings down the cost of the enzyme to affordable levels. We can certainly also regenerate the enzyme or use it multiple times or immobilize it. We've done experiments for that, where we've immobilized the enzyme either covalently or via a biotin-streptavidin interaction. And in that way, used it a couple of times, also in a flow type manner. That is all possible, and I think the number of times we reused the enzyme when we immobilized it was up to 5, but that was just in a small project we ran in our lab, and I'm convinced that we could improve that as well.
Okay, thank you, Leendert. We've got another one on, is the solvent a factor of the speed of ligation? And also, I guess, on a similar theme, what's the best solvent to be used, or is it dependent on the fragments that you choose?
Yeah, that is very much dependent on the fragments that we choose and also the solubility of the fragments. And that's why we're also applying those solubility tags in order to make it soluble in the pH range where we prefer to work at, which is between 7 and 8.5. We mostly use tricene buffer, but we can also work with phosphate buffer. So is that that type of buffer does not significantly influence the speed of conversion. When we start adding organic solvents like DMF or acetonitrile or DMSO, that does influence the ligation speed, because DMF itself can also dock in the active site, and then the enzyme is no longer available for the reaction. So that we see that up to 10, 15 or 20% is okay, but above that, it starts significantly hampering the ligation speed.
Okay, thank you, Leendert. And we have again on a similar theme around the removal of the enzyme at the end and the actual what type of analytical method do you use to verify that that's been removed in the final API.
Yeah, so we mostly use LC. Also for the long proteins, we could just purify them by preparative HPLC. The enzyme itself is 27 kilodalton large, so there's still quite a bit of a difference between the proteins that we synthesize and the enzyme. And in that way, it could be removed. The enzyme also has a his tag, so you could also apply other means of removing that, although there's then, of course, the chance that you end up with heavy metals in your API, which then needs to be removed or carefully checked for. So that's why we prefer to use preparative HPLC to purify. Okay. And I see that there's also something on host cell proteins. Well, the enzyme itself is purified. So prior to use of the enzyme, all the remainder of the fermentation and production of the enzyme, that is gone. And the purity of the enzyme we use is 100%.
Okay, great. So we've got a question on a specific slide, so slide 16. Does the oxoester functionality require some form of activation prior to file substitution?
No, it is already active when docking in the active site. So the other residues in the active site belonging to the enzyme do activate the oxoester. There's no other means, no adjuvant or temperature or whatever required. So it's just we add the enzyme, we add the ester in trypsin or phosphate buffer, pH 7, 7.5, 8, 8.5, and then you get a reaction. You're also not bound to the ester I showed you, which is the glycine variant, the oxoester glycine variant, but you can essentially take the ester variant of all other amino acids that you know of, and even HMBA is an ester. So that there's quite a broad range of esters tolerated by the enzyme.
Okay. And again, two on a similar theme around disulfide bonds. So how do you ensure correct folding of the proteins with the disulfide bonds?
Yeah, so what we do is we just choose the natural folding conditions where like GSH, GSSG in order to fold the enzyme. That is not different compared to what protein synthesis using native chemical ligation or other ligation means require. And that is also very much protein or molecule dependent. So like in EGF, there are many procedures published in the literature which we could use, but also there are examples of folding of more complex proteins that are difficult and that result in some yield loss, but it is very much structure and protein dependent.
Okay, and I guess following on from that as well, why would you use this technology? Why would this technology be better than native chemical ligation or bisenolide selenoester ligation?
Yeah, so one of the answers is that native chemical ligation or selenochemistry requires cysteines or selenocysteines, and in case you have those are not present in the sequence, like the GRP example, you need to design them in and remove them afterwards, which is an additional step or additional steps. And second is the scalability of these technologies is still a bit troublesome. So the thioesters are intrinsically labile, and that is very difficult to scale up, whereas the Peptiligase technology is not. So we use oxoesters, which are also not as stable as amides, but we can work with very stable esters like HMBA, which have been proven at large scale already to give product in very good yields.
Okay, thank you. And just, yeah, two on a similar theme around so what's the highest GMP scale that this has been validated at?
Yeah, so that's the almost close to a kg that this has been produced at. And that's for the API. So the enzyme is produced in tens of grams, and that is sufficient to produce kgs of API. So roughly the enzyme is added to 1 to 10% compared to the amount of API you want to make. So that is the catalytic scale. And
then there's a question on the solvent. So can you use DMSO or another co-solvent to increase the solubility?
Yeah, that is a possibility. DMSO is a bit troublesome, but some DMSO is allowed. It's quite difficult to get rid of as well. But you can use DMSO, but I would investigate how much. Don't expect that you can add 40 or 50% of DMSO, because that won't work.
Okay. And then we've got a question coming in on the chat as well. So have you synthesized glycopeptides using this technology?
Not yet. That's the answer. But so we have synthesized or we have ligated peptide fragments that contain non-peptide motifs. That is what I can say. So you can choose you can think of a PEG, you can think of a lipidated fragment. And in a way, glycopeptide, of course, it's quite a broad interpretation, but we have ligated to proteins that were expressed in a mammalian system, which means that has glycoside trees on it. But yeah, so in a way that we have done that, yes.
I think we've covered everyone's questions. The Q&A is still open if anyone else wants to quickly ask a question. We've got some time left, but I would just like to thank you all for attending today, and yeah, especially for the questions. They were really good. And of course, thank you, Leendert. That was a great presentation. And just to remind everyone, as I mentioned it in the beginning, if you'd like to catch up on the other two webinars in this series, you will find them online on our social media channels and at bachem.com on our website. So if we're done for today and there's no further questions, yeah, once again, I'd like to thank you all and hope you have a great rest of today.
Thanks a lot, Seamus. Thanks a lot all.
Thank you. Bye.